Very good morning to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, today we are going to discuss about unicellular eukaryotes. Those are protists. Okay, so today we are going to complete the kingdom Protista. From this specific kingdom, we have got many questions in the alternate NEET examinations. Like at least there will be one question from this kingdom, because the boundaries of this kingdom are not well defined and they are the common ancestors for all the multicellular eukaryotes like fungi, plantae and animalia. So, Protists, the boundaries of this kingdom are not well defined, and Protista kingdom stays or stands as a common ancestor for fungi, plantae and animalia. Let us look into the general characteristics of Protista. So, here if we see, these are single celled eukaryotes and they are primarily aquatic. Okay, they are primarily aquatic, they are single celled eukaryotic organisms. When they are eukaryotes, they definitely have membrane bound organelles and well defined nucleus, and the ribosomes are ATS type of ribosomes. They have ATS type of ribosomes, wherein the larger subunit is 60S and smaller subunit will be 40S type of ribosomes. And flagella and cilia both act as motile structures in Protista, whereas in Monera prokaryotes the only motile structure that you can see is what beta? Flagella. But here you have flagella and cilia as motile structures, where their microtubular organization will be 9+2 arrangement, whereas in prokaryotes in flagella microtubular arrangement will be what? 9+0, and there the protein which is present in flagella is what ma? Flagellin, and over here it is tubulin protein. Okay, tubulin protein is present in the flagella of the Protistants. Next, nutrition if you have to consider, as we have discussed, these are unicellular eukaryotes and common ancestor for fungi, plantae and animalia. So, here we can have plant-like protists, animal-like protists and fungal-like protists. So, based on the type of Protista, the nutrition will also vary bit. So, here we have holophytic mode of nutrition, holozoic, mixotrophic, saprophytic and parasitic. These are different modes of nutritions in Protista. Next is what? Reproduction. We have two types of reproduction in Protista, asexual and sexual type of reproduction. Asexual includes fission and spore formation, sexual cell fusion and zygote formation. Next is what? So, here as we have discussed, we have three types: photosynthetic protists or plant-like protists. Include what ma? Chrysophytes, dinoflagellates and Euglena. They include chrysophytes, dinoflagellates and Euglena. Second is consumer decomposer type of protists. Those are fungi-like protists. Best example are slime molds. We have two types of slime molds, cellular slime molds and acellular slime molds. Okay, and then animal-like protists, we have amoeboid, flagellated, ciliated and sporozoans. So, these are three different types of protistants. Okay, photosynthetic or plant-like, fungi-like protists and animal-like protists or protozoans. Okay, now let us go in stepwise way. Okay, first is photosynthetic protists. They include Protistian algae. They include what ma? Protistian algae. Major portion, they form the major portion of phytoplanktons. Okay, they form major portion of phytoplanktons and they are the chief producers of the ocean. Okay, they are the chief producers of the ocean. Very important statements beta. Next, so in this, first we have to look into chrysophytes. Chrysophytes include diatoms and desmites. Desmites are commonly called as what? Golden algae. Okay, so chrysophytes include what ma? Diatoms and desmites. Desmites include golden algae. Mostly they are aquatic, which includes freshwater protists or marine water protists. They are microscopic and they float passively in water currents. Okay, they are microscopic and float passively in the water current. Nutrition is holophytic mode of nutrition because they are photosynthetic protists, and they have photosynthetic pigments which include chlorophyll A and B, xanthophylls and carotenoids. So these are different photosynthetic pigments present in the chrysophytes. Cell wall of diatoms is made up of silica, which is indestructible wall. Okay, indestructible siliceous wall. Diatoms have the cell wall which is made up of silica, which is indestructible. They have two thin overlappings. Okay, they have two thin overlappings. Upper we call as epitheca, lower is called as hypotheca, which look like box like appearance. Many a times questions are asked only on the cell wall of diatoms. Very important, ma. So you have to remember the cell wall composition is siliceous, which is made up of indestructible silica. It has two wall layers, upper is epitheca and lower will be hypotheca. Both of them together forms what? Frustule. Okay. Next. So these diatoms, when they get deposited in the ocean bed, they form, the silica depositions will form diatomaceous earth. They form diatomaceous earth. They are used in polishing, filtration of oils and syrups. They are used in polishing and filtration of oils and syrups. Based on the symmetry, diatoms are of two types, centrales and pinnales. Okay, these are centrales and these are pinnales. Centrales are radially symmetrical. That means we can divide this into two equal halves in any of the plane. Okay, we can divide this into two equal halves in any of the plane. Centrales. Pinnales are bilaterally symmetrical. That is, only by one plane we can divide that into two equal halves. So what did we learn, beta? Chrysophytes, whose cell wall is made up of silica, which is indestructible. It has two wall layers, two thin overlappings like soap box, whereas the upper is epitheca and lower will be hypotheca, both of them together called as frustule. And the diatoms deposition, cell wall depositions on the ocean bed will form diatomaceous earth, which is highly useful in polishing, filtration of oils and syrups. And based on the symmetry, diatoms are of two types, centrales and pinnales. Centrales, we can divide it into two equal halves by any of the plane. Pinnales, they can be divided into two equal halves only by one plane, that is bilaterally symmetrical. Next, when it comes to reproduction, it has two types of reproduction, asexual and sexual modes. Asexual by simple binary fission, which is also called as amitosis, as I told you, because there is no involvement of spindle apparatus or spindle fibers. Second is sexual mode of reproduction, that is by gamete fusion. Both the male and female gametes fusion will form oxospores. This oxospore is called as rejuvenatory spore. Remember that. Oxospore is also called as rejuvenatory spore because these oxospores will regain the shape and size of the diatoms. These are called as rejuvenatory spores because they regain the shape and size of diatoms. Fine, ma. Next, dinoflagellates. These are golden brown photosynthetic protists. Most of them are what? Marine only. Mostly they are marine. Photosynthetic pigments include chlorophyll A, C, beta carotene and xanthophylls. Okay, dinoflagellates, these are called as what, ma? Golden brown photosynthetic protists. Most of them will be marine, unlike the diatoms, which are both freshwater and marine. Here mostly they are marine. Photosynthetic pigments include chlorophyll A, C, beta carotene and xanthophylls. Cell wall, if you have to see, ma, it has stiff cellulosic plates on the outer surface. Okay, cell wall is made up of stiff cellulosic plates on the outer surface, and the nucleus, specific and characteristic feature of dinoflagellates, that is mesokaryon, wherein the nucleus or the genetic material is resembling two features, one with prokaryotes and other with eukaryotes. What is that? As we have discussed, the genetic material is membrane bound. Okay, so it has membrane bound nucleus. That is the feature which is in resemblance with eukaryotes. But the division of the chromosomes is not that evident as that it happens in the eukaryotes. Nuclear division shows most of its similarity with prokaryotes. Nuclear division will show most of its similarity with what, Nana? Prokaryotes. Clear, right? Mesokaryon, this is a characteristic feature of dinoflagellates. It will show the nucleus features are similar to eukaryotic nucleus, and the pattern of division is similar to that of prokaryotes. Clear, right, ma? Now, flagella, if you see, flagella, we have two types of flagella, longitudinal and transverse flagella will be present. So, in our NCERT diagram also we can clearly see the longitudinal flagella and the transverse flagella, like how it is moving. Longitudinal flagella will be moving like this and transverse flagella like this. As such, it will be causing a unique movement which is called as whirling whip movement. Okay, so it will be showing whirling whips movement because of the longitudinal and transverse flagella. Till here it is clear, right, Vita? Next, bioluminescence. Bioluminescence is the characteristic feature of that dinoflagellate which is noctiluca, which shows or emits the light. Okay, this is a marine dinoflagellate. And red dinoflagellates, which is goniolax. I have reminded you about goniolax when I have discussed Trichodesmium erythraeum when I am talking about BGA. Trichodesmium erythraeum will make the sea appear red, that is cyanobacteria. Okay, goniolax is a dinoflagellate which again shows the same presence, that is red tides, formation of red tides in the Mediterranean Sea. So, here because of the rapid multiplication or proliferation, they will be releasing the toxins. The toxin name is saxitoxin. Okay, this toxins will be released and they will kill the marine fish, and finally it will result in what? Red tides in the Red Sea, same as such. Okay, so you have to remember these two examples, need not to get confused over here. Next, Euglena, mostly freshwater organisms. Euglena are mostly freshwater organisms. Cell wall is totally absent in the Euglena, but it has proteinaceous rich layer called as pellicle is present. Okay, Euglena doesn't have cell wall, but it has proteinaceous rich layer which is pellicle, and it has photosynthetic pigment over here, that photosynthetic pigment or eye spot which is called as astaxanthin. Okay, photosynthetic pigment is called as what, Nana? Astaxanthin. This will perceive the light. Okay, in the presence of light, it shows autotrophic mode of nutrition. In absence of light, it acts as saprophyte, okay, which is dependent on the other organisms. So, deprived of sunlight, it acts like a heterophyte, but when the heterotrophic organism, when the sunlight is present, it acts like a autotroph. Okay, so it shows both the phenomena, autotrophic and heterotrophic movement. So, that is why the nutrition mode of Euglena we call it as mixotrophic nutrition. And here in Euglena we have one short flagella and long flagella which shows steichonematic type of flagella. Flagella type is steichonematic type. Fine. This long flagella will have some thread-like structures. Okay. So, this is said to be tinsel flagella. Okay. Tinsel flagella. Clear, right, ma? Next is about contractile vacuoles which are present in Euglena, help in osmoregulation, and it has membrane bound nucleus. So, this is about the structural description of Euglena, which is very important. Okay, very important. Okay, next. Mixotrophic mode of nutrition, as I told you. Photosynthetic, in the presence of sunlight, it acts as holophytic mode of nutrition it will conduct. In the absence of sunlight, it is heterotrophic or holozoic mode of nutrition. And here the photosynthetic pigments, if you see, photosynthetic pigments include what? Chlorophyll A and B, xanthophylls and beta carotene. Heterotrophic mode of nutrition, holozoic, this gets nourishment from dead and decaying organic matter. Till here it is clear, right beta? Next, reproduction. There is no sexual reproduction in Euglena, only asexual reproduction is seen. Under favorable conditions, longitudinal binary fission takes place. Okay, under favorable condition, longitudinal binary fission takes place. During unfavorable conditions, cis or palmella stage is observed. During unfavorable conditions, cis or palmella stage is formed, which serves as a means of perennation. Till here it is clear, right beta? Next, fungi-like protists. Best example is what? Slime molds. Slime molds are fungi-like protists. So here if you have to see, they are achlorophyllous saprophytic protists. Okay, they are achlorophyllous saprophytic protists and their locomotion is through pseudopodia, same like amoeba, like how slime molds will be moved by the false feet, those are pseudopodia. Vegetative stage is observed by multinucleated mass of protoplasm, which is surrounded by plasma membrane. So there is no cell wall in the vegetative stage of slime molds. Keep noted, ma. So it is showing multinucleated or cenocytic condition, which is surrounded by the cell membrane. Okay, so it is surrounded by the cell membrane or plasma membrane, whereas cell wall is absent in the slime molds. It just has the cell membrane which is surrounding the multinucleated cytoplasm or protoplasm. Here next is types of the slime molds. We have cellular slime molds and acellular slime molds. Cellular slime molds, if you see, habitat includes, this occurs on humus containing upper layer of damp soil. Okay, humus containing upper layer of damp soil. Somatic phase is just haploid uninucleoid cells, because cellular slime molds, right, each cell is having one nucleus. So it just has haploid and uninucleated cells. Formation of pseudoplasmodium is observed, that is because of the aggregation of the slime molds. Best examples include dictyostelium and polyspondylium. Okay, these two are the best examples of cellular slime molds. Acellular slime molds, if you see, ma, habitat, this occurs in dead and decaying matter. Somatic phase is dead multinucleated plasmodium. Plasmodium is free living thalloid body. Best examples include Physarum and Physarella. Remember the examples of acellular and cellular slime molds, which is very important. Okay. Next, under favorable conditions, this slime molds will aggregate and they form what? Plasmodium. Okay, they form aggregations called as plasmodium, which will grow into the several feet. Here if you see, during unfavorable conditions, plasmodium will give what? Fruiting bodies at their tips. Okay, plasmodium will give what? Fruiting bodies at their tips, which form spores, and these spores are released through air currents. Remember, the vegetative structure of the slime molds is not having the cell wall, but the spores which are being released from the fruiting bodies of the slime molds during unfavorable conditions will have cellulosic cell wall. Remember that. Okay, so spores are having cellulosic cell walls and they are released through air currents. Till here it is fine, right beta? Next, we will discuss about animal-like protists. Okay, animal-like protists. Animal-like protists. These are protozoans. Okay, animal-like protists. These are protozoans. They are primitive relatives of the animals. Okay, they are primitive relatives of the animals. They are heterotrophs. They act as predators and parasites. Okay, they are heterotrophs. They act as predators and parasites. Cell wall is absent, that is why we call it as what? Animal like protists, and their mode of nutrition is heterotrophic only. As such, we have what ma? We have amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. So, if you can see it over here, amoeboid protozoans, they just have false feet called as pseudopodia. They have false feet called as pseudopodia. With the help of pseudopodia, amoeba will show the movement, okay, movement or locomotion. Next, ciliated protozoan, best example is what? Paramecium, okay, Paramecium, which has two nucleus, macronucleus and micronucleus. We have learnt in cell biology, wherein this amongst these two nucleus, micronucleus is involved in reproductive activities of Paramecium, macronucleus is involved in metabolic activity of the Paramecium. Next, flagellated protozoans include trypanosoma and sporozoans, which are non-motile, okay, these are non-motile sporozoans. Best example is Plasmodium. So, here I have given you the complete tabulated form, like we can learn all the 4 types of protozoans comparatively. So, here if you see amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. Habitat includes, okay, fresh water, marine water and moist soil if it is amoeboid protozoans. Flagellated, free living and aquatic. Ciliated, aquatic, fresh water and marine water, this will be the habitats. Sporozoans will act as what? Endoparasites. Next, habitat. Amoeboid protozoans are free living and they are just few parasites. Flagellated protozoans will act as parasites. All the 4 types, ciliated and sporozoans, all the 4 types will show what mode of nutrition? Parasitic mode of nutrition only. Locomotion. Amoeboid protozoans will have pseudopodia, flagellated will have flagella, ciliated will have cilia and sporozoans, there are no locomotory structures. Amongst flagellated protozoans and ciliated protozoans, which will show the fastest movement? If that is the question, what will be your answer beta? Ciliated protozoans will show fastest movement. Next, distinct features. Distinct features about amoeboid protozoans, marine forms and they have silica shells over it. Ciliated protozoans, they have cavity or gullet for food transport. Sporozoans include infectious spore-like stage. Okay, these are the key features or distinct features of 4 different types of protozoans. Now comes the major segment, that is examples, which are very important. Amoeboid protozoans, best examples include what ma? Amoeba and entamoeba. Flagellated protozoans, trypanosoma is the best example. Ciliated protozoans, Paramecium. Sporozoans, best example is Plasmodium. Okay, so note down all the examples as well. So with this we have finished out the protistants. Okay, 3 types: plant like, animal like and fungi like protists. And in our coming class we will be discussing completely about fungi, which is the heart of this chapter, to be frank. Okay, this is the heart of the chapter, fungi. From fungi definitely we can expect one question in the final NEET examination. Mostly the questionnaire for fungi will be arised from the reproduction of fungi. Okay, so we will be dealing that in the most clear way. So don't miss out the next session. See you tomorrow. Thank you.